Welcome to this lecture on cell biology. Today, we are going to explore one of the most fascinating and crucial communication systems inside your body, the cyclic AMP and protein kinase A pathway. Imagine if you could trigger a massive, coordinated response across an entire city just by knocking on a single door. That is exactly what this pathway does for your cells. We will build our understanding from the ground up, starting with a simple analogy, and eventually uncover the mathematics that make this biological amplification possible. Let us dive in. To begin, let us imagine you are the manager of a massive, bustling factory, but you are locked outside. You need to speed up production, but you cannot walk through the doors. Instead, you knock on the window to alert a security guard. The guard immediately pulls a fire alarm, and that loud ringing echoes across the factory floor. Hearing this, the sleeping floor supervisors wake up and shout instructions to the workers. In this analogy, you are the external hormone. The guard is the cell receptor. The alarm is a molecule called cyclic AMP, and the supervisors are protein kinase A. This beautiful relay system allows a message from the outside to cause massive action on the inside without the original messenger ever stepping foot indoors. Now that we understand the factory analogy, let us look at the actual biological mechanism. In the formal language of cell biology, an external signal, like a hormone, is called a primary messenger. When this ligand binds to a G protein-coupled receptor on the cell membrane, it activates an enzyme called adenylyl cyclase. Think of adenylyl cyclase as the security guard pulling the alarm. This enzyme takes ATP, which is the standard energy currency of the cell, and converts it into cyclic adenosine monophosphate, or cyclic AMP, by removing two phosphate groups. Cyclic AMP then floods the inside of the cell, acting as a secondary messenger. It carries the signal deep into the intracellular space, where the original hormone could never reach. With cyclic AMP flooding the cell, this brings us to the next crucial step: activating the floor supervisors. The primary job of cyclic AMP is to find and bind to an enzyme called protein kinase A, often abbreviated as pKa. pKa is a kinase, meaning its sole purpose is to attach phosphate groups to other proteins, a process known as phosphorylation. In its resting state, pKa is completely inactive because it is bound to regulatory subunits that act like a physical lock. When cyclic AMP binds to these regulatory subunits, the lock opens. This releases the active catalytic subunits of pKa, allowing them to go forth, phosphorylate target proteins, and fundamentally change the behavior of the cell. Understanding how pKa is unlocked leads us to an important question. Why does this matter for our survival? This pathway is the fundamental mechanism behind how our bodies respond to stress. For example, when you get scared, your adrenal glands release adrenaline. This binds to receptors on your liver and muscle cells, triggering a massive spike in intracellular cyclic AMP. The resulting activation of pKa leads to the rapid breakdown of glycogen into glucose, giving your body the sudden burst of energy needed for the fight or flight response. Without this secondary messenger system, your cells would completely ignore hormones. Furthermore, many pharmaceutical drugs, including asthma and heart failure treatments, specifically target this pathway to artificially control cyclic AMP levels, proving its vital role in human medicine. Before we move on to the mathematics, let us see this entire process visually. Here you are looking at a schematic of the cell membrane. On the far left, notice the adrenaline molecule binding to the G protein-coupled receptor on the outside. This triggers the adenylyl cyclase enzyme right next to it. Look at the arrows showing ATP being converted into cyclic AMP. These cyclic AMP molecules then travel to the inactive protein kinase A complex in the center. You can clearly see how the binding of cyclic AMP causes the regulatory subunits to detach, freeing the blue catalytic subunits. Finally, on the far right, these active subunits attach a phosphate group to a target protein, executing the cell's final response. Now that we have visualized the pathway, let us understand the sheer power of this system through the mathematics of signal amplification. Every single step in this biochemical cascade multiplies the effect of the previous step. We can represent the total number of final activated product molecules using this simple multiplication formula. The variable P represents the total number of final phosphorylated target proteins. L stands for the initial ligand molecules, like adrenaline. C represents the cyclic AMP molecules produced by adenylyl cyclase for every ligand. We multiply by the fraction 1/4 because it takes exactly 4 molecules of cyclic AMP to activate one single molecule of protein kinase A. Finally, T represents the number of target proteins each active pKa can phosphorylate. If cyclic AMP increases, the final response grows exponentially. Let us see this formula in action with a concrete example. Imagine a single molecule of adrenaline binds to a liver cell. This single event causes adenylyl cyclase to produce 4,000 molecules of cyclic AMP. We also know each active pKa can phosphorylate 100 target enzymes. We need to calculate the total number of phosphorylated targets. First, we determine how many pKa enzymes are activated. We take the 4,000 cyclic AMP molecules and multiply by 1/4, giving us 1,000 active pKa enzymes. Next, we take those 1,000 active enzymes and multiply them by the 100 targets each can phosphorylate. The final answer is 100,000 phosphorylated target enzymes. A single external molecule resulted in 100,000 activated enzymes inside, providing a massive biological response. This massive amplification leads us to another practical scenario. What if we need to work backward? Sometimes researchers need to know exactly how much cyclic AMP is required to achieve a specific level of pKa activation. We can express this relationship with a simple formula. C equals S times R. Here, C is the total required cyclic AMP molecules. S represents the desired number of active catalytic subunits we want to release. R is the biological ratio of cyclic AMP molecules required per catalytic subunit. Because a single pKa holoenzyme requires 4 cyclic AMP molecules to release 2 catalytic subunits, this ratio R simplifies to 2. This means for every 1 subunit you want, you need 2 cyclic AMP molecules. Now that we have a ratio formula, let us apply it to a research problem. A researcher is studying a mutant cell line and determines she needs exactly 5,000 active pKa catalytic subunits to observe a visible change in cell behavior. The cell currently has no active pKa. How many molecules of cyclic AMP must be synthesized? We start with our given requirement of 5,000 subunits. We know our biological ratio is 2 cyclic AMP molecules for every 1 catalytic subunit. By multiplying the 5,000 required subunits by our ratio of 2, we find the total requirement. The cell must synthesize exactly 10,000 molecules of cyclic AMP. This demonstrates how tightly regulated the system is, allowing the cell to precisely tune its internal response. Having successfully used the ratio formula, let us take a moment to formally derive where that ratio comes from biologically. We are setting out to prove why the multiplier is exactly 2. We start by looking at the physical structure of a single pKa holoenzyme. It is built from exactly 2 regulatory subunits and 2 catalytic subunits. Biological studies show that each individual regulatory subunit requires exactly 2 molecules of cyclic AMP to change its shape and unlock. Therefore, to unlock the entire holoenzyme, we multiply the 2 regulatory subunits by 2 cyclic AMP molecules each, giving us a total requirement of 4 cyclic AMP molecules per holoenzyme. Continuing our derivation, we now look at what happens after those 4 cyclic AMP molecules bind. Unlocking the holoenzyme releases its payload, which is exactly 2 active catalytic subunits. To find the ratio of cyclic AMP per single catalytic subunit, we divide the total cyclic AMP used by the number of subunits released. We substitute our values into the fraction, giving us 4 divided by 2. Simplifying this fraction yields our final result. Exactly 2 cyclic AMP molecules are required for every 1 catalytic subunit released. This mathematical proof directly reflects the physical biological structure of the enzyme, ensuring our cascade calculations are perfectly accurate. We have covered a lot of ground today, so let us summarize the most important ideas. First, remember that cyclic AMP is a secondary messenger. It acts as a loud internal alarm bell that allows cells to respond to external signals without letting those signals inside. Second, cyclic AMP does its job by binding to and unlocking the regulatory subunits of protein kinase A, waking up the cellular supervisors. Third, this entire pathway is a brilliant biological relay system that amplifies a tiny external whisper into a massive, coordinated cellular action. Finally, by understanding the mathematics of this cascade, we can precisely calculate how our bodies regulate energy and respond to stress. You now have a solid understanding of this vital biological pathway.